Hi, everyone. It's a pleasure to be here today. My name is Eric. I'm a postdoc in the Vento Lab at the Sanger Institute. And today I'm going to show our latest tool to study metabolism. As we know, metabolism is central for cellular life in general, and it's often defined as the set of reactions within an organism that allow the organism to sustain different functions and sustain life. And the way that metabolism can help to perform different functions, first is by converting nutrients into the different building blocks that are necessary for other processes, as for example, amino acids, lipids, nucleotides, and energy. And with that, with those set of building blocks, then cells can synthesize different macromolecules like DNA, membranes, and proteins, which enable the performing of different cellular processes and functions. Because of this reason, as you can see, metabolism is key, and studying it is very important to have an idea of different phenotypes and things happening inside the cells. Normally and ideally, the way of studying metabolism would be using metabolomics, but in most of the cases when we want to go into the single cell resolution or even a spatial resolution, the access is very limited for most of the researchers. And that's why transcriptomics is a nice alternative, not the perfect one, but could help to understand what's going on with this kind of resolutions. And one way of doing that is by relying on prior knowledge from genome scale models, where we have in these big networks of metabolism different reactions, the metabolites that are participating in these reactions, and even mapping the different enzymes involved in these reactions and the genes that are associated to those. And with that, we can define something that is called metabolic tasks, which is basically a set of reactions. We could be described as a pathway, but it's even more specific because it describes the conversion of a given metabolite into another final metabolite. And since we know all the reactions from the genome scale model and the enzymes involved in catalyzing each of these conversions, then we can track the different genes encoding these reactions. In some cases, could be complex proteins for one enzyme or just a simple protein that is the enzyme itself. So for that, we developed a tool called SysMod, which basically takes this information from the prior knowledge in the genome scale models and a database of metabolic tasks with the different reactions described. And we can use single cell data and spatial data as well to predict different metabolic activities at the level of the metabolic task, as you can see here. So with this activity that we predict with our tool, we can then perform a different set of analysis, including identification of metabolic markers at the level of some group of cells, the spatial analysis to detect some patterns maybe in the tissue, compare different conditions, even extend this to understand maybe cell-cell communication using metabolites, and extract other patterns as temporal patterns, for example. The way that the tool works, I'm not going to go into the details in the calculation, but just to give you an overall idea how it works, we start with our gene expression matrix, and we end up with another matrix with metabolic score for the activities of the different metabolic tasks. But in between, we have an intermediate set of other calculations. So first, we need to make sense of the gene expression, and that's why we get what is called a gene score, which basically describe across all the metabolic genes, the one encoding the enzymes, whether they are above or below a threshold to say potentially this enzyme or gene could be metabolically active. And the way we define this threshold is basically inspecting the whole distribution of the metabolic genes, and then we can have an idea which range of value they could move, and then maybe say, okay, maybe from here could have some metabolic activity. Once we have these gene scores, we define the reaction activity. So basically, we map all these genes into different enzymes, and we know which enzymes are catalyzing the different reactions. And a way of doing that, in some reaction, we have multiple enzymes catalyzing the same reaction. In other ones, just one, for example. But the way of doing that is identifying the maximum activities among the different enzymes. And for the enzymes, also, we can have different subunits when we have complex of proteins. So we also need to identify the bottleneck of those complexes because we need all the the subunits to be present and active. And once we have that score, we finally aggregate this reaction score into the metabolic task, that is the set of different reactions, by using an average also that considers some specificities of the different genes that could be involved in multiple other reactions. So this is like the big picture of how the method works. And since in Sanger we analyze generally large single cell and spatial atlases, first thing we did was trying to see whether our tool is power enough, the power it has is enough to analyze millions of cells, single cells in this case. And also we need to make sense of some biology in the different cells. So we studied the whole human cell atlas, in this case about 30 million of single cells across different organs. And I'm not going to go into detail all the single cell, of course, it's impossible, but just to summarize, we predicted for every metabolic task in our database and metabolic activity score. And then here I'm just summarizing the average across the different organs. So each bar basically is a metabolic task. And the level of the bar is the average across the organ. The color is the major category of these tasks. And you can see some of them seems to be highly active across most of the organs because they have a higher average, but other ones have a lower average. And when inspecting this, especially, we realize that some of cases, as the one here, even though they have a low average across organ, they are very specific to a few or maybe one of them. And as you can see here, the same representation now per organ. We see, like, for example, glutathione synthesis is very specific in the eye, and the same with adrenaline production is very specific in the adrenal gland. And since we know the single cell activity, we can map all the single cells we have in every organ and see the activity and identify maybe which group of cells are the ones performing that conversion. And we can see in the eye the lens fiber cells, which are important in this case to synthesize glutathione and help with the oxidative stress of light in that case. And in the adrenal gland, we can see the chromatin cells that are the one synthesizing the adrenaline, which is biologically relevant. So just to summarize what we did with the Human Cell Atlas here, we also put available a platform, sysselfie.org, where users can upload their own analysis with their own dataset and also explore our prediction in the whole Human Cell Atlas. Also, there are some tutorials available to perform different set of analysis. And most importantly, you can inspect interactively the different scores and the ones that predicted in the Human Cell Atlas. So with that, we show that it's scalable. We can explore large data sets. But in the Bento lab, in particular, we are interested in studying the female reproductive system. So we focus here in the endometrium, which is a lining of the uterus. In this case, this tissue is very particular because it's highly dynamic. It changes a lot across the menstrual cycle. And it responds to hormonal changes, which make it susceptible, for example, to cases like obesity or diabetes, where we have different changes, and that could leave some reproductive dysfunctions. So by using an atlas previously published in the lab of the endometrium, we have different cell types across the menstrual cycle. So we use SysSelfie to predict metabolic activities. And at first, sorry for the size, I think it's not visible to everyone, but basically we have here different metabolic tasks and the different cell types, and we try to identify some metabolic markers from these predictions in the cell types. And in the left side, we can see some of the epithelial cells in the endometrium, or stromal cells. And in red, we got interested to evaluating their specificity in the space as well. So we use a Visium data set previously generated in the lab as well, where we have the different epithelial cells in the lumen or the glands, and stromal cells as well. And by using the ones in red, we can see some of the cases that were predicted to be important for the glands and the lumen. They are consistent with the spatial location of the cells. Other cases more in the lumen, like arachidocyte production, and the ones that were participating with the stromal cell also are highly active in different areas of the tissue. So now that we have an overall idea of the healthy tissue. We also wanted to know a bit more what's changing maybe in diseases cases in the endometrium. So one disease we explored was endometriosis, which is characterized when an endometrial cells start growing outside the uterus, and that leads into different lesions in different tissues and organs. And in even more severe conditions, women can go in their infertility and some bleeding problems. So our first thing we wanted to understand was whether there is some changes in the potential in the uterus and the endometrium located in the uterus, not in the lesion outside of it. So we compared the ectopic endometrium of women with and without endometriosis. With every row here, we have a different cell type. In the x-axis, we have the effect size of these changes, and every dot is basically a metabolic task. So just to simplify it, maybe this could be seen as a volcano plot, like a flattened volcano plot per cell type. And we can see that many changes are happening in the epithelial cells during endometriosis. But the most remarkable finding we saw that only among the immune cells, only macrophages are the ones changing here in some metabolic tasks. And this is consistent with previous observation in our lab where cell-cell communication seems to be contributing to a pro-inflammatory environment with the macrophages here playing an essential role. And when inspecting some of the changes in macrophages, we identify some cases that produce metabolites that also favor this pro-inflammatory microenvironment. And epithelial cell, we have a lot of changes, including amino acids and different carbohydrate metabolic tasks. So we further inspected a spatial data set now in a ectopic lesion growing outside the uterus, like in the peritoneal lesion. And we mapped the different cells that could be important. So we found macrophages surrounding the lesions, endometrial epithelial cell also growing in the lesion. And when mapping some of these metabolic tasks, we saw some enrichment as well in these lesions, which suggests that potential in the endometrium also could be involved in different lesions and in the development of endometriosis. And the other disease we inspected was endometrial carcinoma, which is a case where a metabolic rewiring really drives this disease. And especially, for example, in women during postmenopause with obesity, there is an increase of estrogen production, which normally postmenopause, there is a drop, but here, because of obesity, the adipose tissue produces more. That favor more proliferation of epithelial cells and generates a whole metabolic reprogramming and leading potentially to tumorigenesis. So it's a risk factor in this case. We evaluated a spatial transcriptomics data set that publicly available with regions annotated with malignant and normal cells. And one thing we first did was trying to find maybe some spatial organization of the metabolic activities. So just by running different algorithms to identify this spatial organization. For example, we found the typical hallmark of cancer, that is the lactate production, enriched in the malignant regions, and also mevalonate synthesis that could contribute to epithelial cells proliferation. Statistically significant, as I mentioned, given the regional annotations. And also we wanted to understand what happened during early stages of this cancer. So by using a gene set previously defined in other work for the early stages of endometrial carcinoma, we computed a tumorigenesis score based on the gene expression of these genes. And you can see here some of the malignant areas have a higher score, potentially being the ones involved in the early stages of this cancer. And just by simply using correlation with our metabolic activities predicted with our tool, we found one of the tasks involving the tryptophan metabolism to be highly active in this region that could be tumorigenesis regions. And when we localize this task into the different pathways it participates in the kynurenine pathway. And you can see here, kynurenine is derived from here. But this metabolite in particular also is important for cell-cell communication. So since this task could be active, we got interested whether this could be involved in the communication. And just by using the colocalization with the receptor, we found that the high potential of communication also is consistent with this tumorigenesis score, and also significantly important. So just to summarize, with this tool now we can add another layer of biology when using transcriptomics data. We can now easily study metabolism, not only other processes that normally are done with this kind of data. And because of the way we interpreted metabolic tasks, we implemented metabolic tasks, it's easy to interpret. We can analyze large data sets, and as I show, it's important to study healthy tissue and also during disease, and help also with hypothesis generation to ask new questions. So the work is available on bioRxiv if you are interested. And I would like to thank the whole lab, especially Roser and Luz, that were important to framing this work, and the collaborators, Nate, because of all the background of the metabolic modeling part as well, and the funders. With that said, I would be happy to take any questions.
Thank you. I have some questions, even that we have time. So your approach is data projection without simulation. I mean, you do not exploit the stoichiometry, the mass balance constraints. Beyond the computational reasons, do you have any specific motivation for not using flux balance analysis and just
yeah, well, as you said, one of the main reasons is basically that, the computation, how long it could take us to explore millions of cells, but also, given that this gene expression, sometimes a bit tricky to define gene expression with constraint of fluxes. It's not, there are some methods, of course, that work well for some cases, but just making those assumptions maybe is not the best approach, but could be useful to extract other kind of information, maybe if you are interested in the metabolic fluxes.
And also when you compute your score for your task, you are averaging the activity, the reaction scores, right?
Yeah, yeah.
So there you can lose a lot of information because your average is sensitive to
exactly.
outliers, and maybe you have like, and gene expression can be pretty inconsistent within a pathway, so maybe one gene that So I was wondering why you do not use like some more standard but more sophisticated approaches like gene set enrichment analysis, so you can try to reach your pathway against.
Yeah, but one problem with that, and actually this is work that we could do later for the tool. You assume that all the genes contribute the same, but here, since we are using the GPR rules, we are kind of making things.
I mean, using the GPR
yeah, I see.
And use them instead of the gene levels and try to reach reaction sets, like moving from gene sets to reaction sets enrichment. So maybe you can have some p-value and some more statistical power as compared to a simple average.
That's something that could be done for sure, but when we were developing this, we were also thinking the improvement that we could do. So one improvement that could be done is taking also the whole pathway and see maybe if we have the directionality of the pathway and whether some steps that could be bottleneck are missing or not. So ideally, we could add that information as well in the way we compute our score. So by using the enrichment, we cannot do that. So, I mean, not exactly. No, probably not the directionality, but if you have the pathway, starting with one metabolite and ending with other, then you can see the different steps and say, okay, maybe from our scores, we are missing this step. So the pathway is incomplete. So we can add that to the calculation, and we're hoping to add it eventually.
Okay. So questions from the audience?
Yes.
Okay. Do you mind reach the microphone? Otherwise, the people that
No, it's a very short question. You mentioned endometrial cancer. So endometrial cancer is also one of these not so numerous cancers that display mutated phenotype. And with a very high number of mutations caused by mutations generated by a mutation in polymerase epsilon. So is there any reason, metabolic reason, in the environment in which endometrial tissue is residing for this particular phenomenon.
So can you repeat the last part because I cannot hear you with
Okay,
the.
well, so is there any reason, metabolic reason, the environment in which the endometrial tissue resides for this type of mutations to occur? I mean, hypermutated polymerase epsilon mutations.
Yeah, that's a good question. I'm not an expert in that kind of cancer, to be honest, but I think depending on some changes in like cells communication and other things that contribute to the tissue microenvironment, that could lead to some intracellular changes as well. And maybe because of those activities, there is like a selective pressure maybe that drives some mutations.
20% of endometrial cancers that display this phenotype, much higher than any other.
Thank you.
Okay, if there are no other questions, let's thank again our speakers.